session will be recorded. Right, let me share my screen in presentation. Okay, hi everyone. Welcome to today's webinar. My name is Stella, USP Strategic Customer Development Manager for Southeast Asia. So again, a couple of housekeeping information for today before we start. Now, if you cannot hear us at this time, please be sure to join the audio conference by selecting your name and then clicking the audio button as shown on the screen. Set your computer or any device that you are using as the audio output. Okay, I hope the audio works well for you. Next, very important, if you have any questions that you would like us to address today, please feel free to key them into the Q&A tab on the right-hand portion of your computer screen. Please do be sure that the questions is sent to all panelists so that myself as well as our speaker for today will be able to view them, and we will do our best to address as many questions as we can for today's Quality Hour webinar. All right, good morning and good afternoon, good evening to everyone. Welcome to our 16th Quality Hour. So Quality Hour is a series of webinars developed by the team here in USP Asia Pacific to stay connected with you. For today's session, we will have Donald Charles to discuss a little bit more about proficiency testing, and then I will give you an update from USP, namely some of the new reference standards and compendia updates in the month of October and November. As always, before we end the session, we will have some time to take on some questions, live questions as well as questions that were sent in during the registration. Again, please feel free to type in any questions that you may have during the session in the Q&A tab. If you cannot find the Q&A tab, the chat box will work as well. Now, without further ado, I will hand over the time to Dr. Donald Charles, Senior Manager, Proficiency Testing Program. Dr. Charles has been with USP since 2012. He just shared with me that information, and he has many years of experience in laboratory quality management system and knowledge in numerous ISO standards. Speaking from the USA Today Live, he will be sharing more about proficiency tests during this 35-45 minutes session with him. So without further ado, I will hand over the time to Dr. Charles, and then I will pass on Dr. Charles the presenter rights. Yes, you should have control, so.
Can you see my screen?
Yes.
Excellent. Good afternoon, good evening, good morning, everyone. My name is Donald Charles. I am the Senior Manager for Proficiency Testing at USP. As you know, USP has a variety of products. Their primary focus is reference standards and documentary standards, but they also have products and services in impurities, consulting, and verification. USP has decided to offer a new product and service, which is proficiency testing. Today, we're going to go through an overview of proficiency testing, how USP is a new player in town, how to order, report, and how it's different from a reference standard, how it all ties in together, marketing, critical dates, and customer service activities. Hopefully this overview will give you information and guide you on how proficiency testing works here at USP. So if you're in the lab, if you're ISO accredited, or if you're trying to build capacity, look at training, or continuous improvements, proficiency testing may be an opportunity for your lab and your participants or other customers. If you're not aware of what proficiency testing is, it is evaluation according to ISO 17043 of an established criteria. Proficiency testing uses schemes of studies and also refers to an evaluation of competency of an analyst in a lab with specific methods and materials. A scheme or study is a combination of test items. As you know, a test item can be API, bulk, or living organism, and methods evaluated at a specific time. What is proficiency testing used for? It is used to measure performance and the ability of performance between two or more participants or laboratories. It provides accurate and precise results. You want to make sure that individuals performing studies can get the same results precisely and repetition. It provides similar or same materials. In a proficiency study, there are multiple participants or labs testing the same material within the same window or time. And it is in accordance with pre-existing or predetermined analytical protocols, conditions, or requirements. So the analytical protocol and methods that we use to build this program were the general chapters and monographs at USP. A lot of individuals ask us what are the laboratory benefits, and in general, amongst PT providers, it demonstrates competency with a third party. So this material will be provided to your laboratory or participants outside of your lab. It helps you manage risk and early warning. If your staff has challenges doing a particular test, PT can be used as a control process or a training process to evaluate and monitor risk early. It is an independent evaluation of the performance of tests and methodology in your lab. It can be used for educational tools for onboarding or general training for continuous improvements. It meets the requirements of laboratories that are ISO 17025, 43, 15189, and other accreditation activities, or it meets the requirements of your regulator or internal processes. The lab is able to compare results with laboratories around the world. Excuse me, there's something in my field here. It ensures customers' confidence in your laboratory's results. If your laboratory is accredited or WHO pre-qualified, customers are ensured that the confidence of the data in your lab is correct and the results are valid. Also, PT allows you to identify problems early in areas of improvement in case there is corrective action or preventative action. USP program, customer benefits. We have an in-house manufacturing process with an established provider, which is NSI. We leverage trusted USP methods. So as you know, USP has reference standards and documentary standards that build on USP methods. We have the best-in-class scientific expertise under our compendium method. And we have highly trained experts that will respond to your technical activities and technical support, questions, or complaints. USP is not a first to market, but we are a new player in town. As a new player in town, we have gone through a strategic alliance between USP and our collaborator, NSI Lab Solutions, to develop and market PT schemes. NSI Lab Solutions is a highly reputable player in the PT market industry. They are accredited ISO 17043, which is for PT providers, ISO 17034, which is for reference material producers, ISO 17025, which is for testing and calibration laboratories, and ISO 9001. The USP team, along with NSI, developed and identified specific schemes and methods and operational plans. We also market and sell our products for basic as well as advanced laboratories. Together, USP and both NSI have certain synergies. At USP, we have test methods that are globally recognized and valid. We have an unparalleled reputation in the quality space, especially with the GMP manufacturers, micro and other clinical and biological spaces. And USP has a depth of scientific expertise known around the world. NSI Lab Solutions is an accredited provider with extended experience in manufacturing of PT materials, both API bulk and finished products, and they are responsive in their customer service and customer details when it comes to ISO 17043. Now, a lot of labs and a lot of customers ask us, how do we compare against our competitors? Well, as this slide shows, our three major competitors globally are Sigma Aldrich, LGC AXO, EDQM, and a host of other ISO 17043 providers. But USP is program-based and a standard-setting organization. We are not a chemical company trying to be a PT provider. We provide schemes that are fit for purpose, so we cover GXP industries, GLP, GMP, GTP, GCP. Our primary focus or strength, though, is in the quality of food, drugs, and medicine. And that's where our strength lies, especially within our reference standards and our compendial documentary standards. USP and NSI have multiple ISO accreditations and certifications, as do some of our competitors. Our technical expertise, though, lies directly in our USP general chapters and monographs that support multiple industries. And our primary business is focused on those documentary standards, reference standards, and reference materials that support multiple industries and multiple laboratories. You may ask yourself, how do we select our materials and our criteria? Well, we look at multiple methodologies within the USP and is harmonized against the USP and other compendial methods. We also make sure that the material that we select is able to be tested and is suitable, has a strong morphology, and is robust and fit for use or purpose for our particular participants in the lab. And we ensure that the hazards are low or the quantities of hazards in our materials are low. Secondly, we check for the homogeneity and stability, as all PT providers do. We ensure that the physical properties meet the requirements. We ensure that the material or organism is safe and non-toxic and non-lethal. All of these combined help USP select the materials and criteria for our schemes or studies. There are two ways to order materials from USP. The majority of our overseas customers work through distributors, or you can order material directly. But if you don't know how a PT works, it's 4 easy steps. A customer will look up the provider or select a provider, order a scheme. That scheme is either sent directly from the provider or through a distributor. That physical sample goes back to the customer. A customer must have time to analyze the material, develop the results, and submit the results back to the PT provider. The PT provider will then review the results and generate a report. That report is sent back to the customer, which they will use either for their accreditation, qualification, regulator, or internal policies and practices. Remember that test results may only be used and entered by the end users. So when ordering through distributors, make sure that you have the proper documentation from distributors in order that the end user can test the material. Currently now, based on this slide, USP has 23 schemes and offerings in the pharmaceutical space. We have another 4 additional schemes coming up in round 2. Our schemes consist of materials based on bulk material and finished products, along with microbiological material. The schemes are based on the general chapters and USP, or harmonized against general chapters in the USP. These tests were selected based on criteria for either basic lab testing or laboratories to test advanced items, finished goods, and materials. We want to focus on a broad market, but at the highest level, we focus on 5 specific areas. They are the GMP manufacturers first and foremost. We also focus on OMCLs, which is Official Medicines Control Labs, in support of regulators. We look at CROs, commercial labs, pharmaceutical cosmetic organizations, food organizations, and contract drug manufacturers. All of these labs at the highest levels support quality control, proficiency testing, or a means of improving lab performance through either interlaboratory testing or proficiency testing. These laboratories, though, internally have a specific audience that we target. And we try to target department heads, laboratory directors, lab managers, and quality managers. They are the decision makers that will provide the support to the laboratory, whether to do a PT test, when to do a PT test, and when to schedule the performance of a PT test. The QC managers, principal investigators, and laboratory technicians are the individuals who implement the test, drive analysis, develop the results, and submit the results back to the PT provider. Now, one may ask how to order it. This is not a marketing informational. This is more of a PT technical informational. But I want to explain to you about the customer experience. On the USP website, if you go directly to the USP website, there'll be an area which is listed as products and services. Or you can go to our collaborator, NSI website, and order directly, as we have a portal for the USP PT program on their website also. Because this is a USP training, though, we'll focus on how to navigate through the USP site. If you go to products and services, you go and see in the services section, proficiency testing program. That will lead you to an area that gives you full details on the new USP program, general overview of the program, and how USP is committed to global public health and building trust and quality in medicines. And this service aids in those areas. On the page, you also will see a Learn More. That Learn More will take you also directly to the NSI ordering page for the customer experience. As you go to the NSI customer page, you'll see a section called USP Proficiency Testing. And as previously stated, it will demonstrate or display 23 studies or schemes directly related to compendial and general testing and general monographs and general chapters. As you scroll through the system, it will display the item, it will display the round. Currently, USP is supporting 3 rounds in September, January, and April. As you scroll through the sections and the customer experience, it'll have a product detail, USP schedule, and an MSDS for safety. The product detail describes how the laboratory will perform the assay, what is in the material, the controls, and what monograph or what general chapter to reference. We also have related products of interest if the customer cares to add additional tests to their schemes or sections. We also supply a SDS, or material safety sheet, or data sheet, for those individuals who are ordering the material, who want to know how to handle the material, the hazards of the material, and how to safely transport the material from one site to the next or one lab to the next. So as you can see, we have our studies in 9/21, which was September. We have our next study in January 2022. We have another study in April of 2022. As we scroll through the customer experience, you can add the material that you're interested to your cart. You can view your cart to ensure that the material that your group has selected is in the cart, and you can apply your information. which will show the subtotal on our e-commerce page. You can check out. Now, the checkout is important. You can have the material shipped directly to you, or if you are using a distributor, the distributor can ship that material directly to your lab via the distributor. What is most interested in most labs is the final report. Remember that USP is going to provide the participant a final report to have the objective evidence to use either for ISO accreditation, pre-qualification, internal processes, or regulation. A PT provider must have a means for a participant to order the material and a means for the participant to report the data. On our USP platform and NSI platform, there's an area to log in, submit your password, and select your study when reporting your data. Each individual lab is given a unique ID and information. So therefore, the laboratory has a means to report the data independently. The participant will enter the information, put in a specific lab code, and locate their study. They will enter the required information and submit the data back to the provider for evaluation. And you will have to repeat this process for each study. A notification will return to the participant saying that the provider received your information within the specific time and round that is required. After you've submitted your round and the results, a PT provider must give you a final report which they have evaluated. In this report, submitted by the participant, it will provide a laboratory code only known by the participant, information related to the study, shipping information, closing information, and the date that that information was submitted and printed. Currently now we provide two types of reports: a quantitative report, which shows the analyte of interest, general details, but the most important area on this report is a Z-score. That Z-score determines whether an analyst has received an acceptable data or passed through tests, and who and when passed the test. Remember that the closer you are to the PT provider's internal results, the better your Z-score. We also provide qualitative tests. So if you are working in the micro field, or clinical field, or biologic field, you may receive a qualitative test. Once again, it shows the analyte of interest, whether the results are passing or failing, and the participant and the results. Finally, if you are ISO accredited or WHO pre-qualified, your accreditation body or your assessor may look for values on how the criteria of evaluation was made, who did the evaluation, whether your PT provider was accredited, and who reviewed it. There's also opportunity to have questions answered from your provider, and also the report has to be confidential between the provider and the participant. This is also a time on the report, if you disagree with the results or the results generated, you can complain to the provider, and the provider must reply to you in a reasonable amount of time. Now, we want to show you that the difference between a PT material and a reference standard, and how PT and the material in PT is not a reference standard. A proficiency test and the studies involved involve the evaluation of a laboratory's performance or individual in the laboratory's competency against other laboratories. PT can be used for risk assessments, quality, corrective action, preventive action, or responses to your accreditation body or your qualification provider. While reference standards are predominantly used for API or bulk identification, or to implement a quality control process through analytical means in support of releasing materials or registering materials. How it all ties together. If you ever have purchased reference standards or have used the documentary standards, reference standards are associated with the product the majority of the time, or a finished product. And I'll provide an example here. This reference standard below is associated with acetaminophen, and you may be testing this material in a tablet form or capsule form. Whether you're using USP's monograph or other compendials, you must identify the material through a performance test or assay. Those tests are associated with the monograph, and within those monographs may be a general chapter. Those general chapters may reference a technical instrument or a piece of equipment that allows you to perform the assay. We built our PT schemes around our general chapters. This means that an analyst must understand the products, understand the monograph, understand the general chapters, be able to use the instrumentation affiliated with that general chapter, and be able to perform the method as designed or as read from either the compendial method or instructions provided from the PT provider. PT provider, you must be aware of critical dates, and we must inform customers of these critical dates. Currently now, USP is going through round 1 of our study, which started in September and concludes in December. Those are routine studies, and the routine studies are also in round 2 and in round 3. Round 2 begins in January of 2022 and ends in March. Round 3 starts in April and ends in June. As of December, we also have PT Express. So if a laboratory needs to do a PT quickly, we can provide that material directly, and you will not have to wait for the results at the conclusion at the end of the round. We also have 4 new studies or new schemes coming in our round 2 in January, which mostly focuses on micro testing. Here's something important to know. When we have a round, there is a time where you can pre-order and review the information during the study. Remember that as a PT participant, you must order your material in time, whether it goes through direct shipment or through a distributor, in order for you to test the material within the laboratory, review the data within the laboratory, and submit it back to the provider for the provider's review. Our current round 1 ends on December the 10th at midnight, U.S. Eastern Standard Time. The report will not be generated for all participants until 10 days later, which is December 20th. So if you're conducting or participating in a PT routine round, you will have to wait for the end of the round to receive your reports. If you decide to do PT Express, the reports come within 48, 72 hours of completion and submission back to the PT provider. Some of the things to know. Domestic and international shippings occur 72 hours after orders. Participants must report the results midnight Eastern Standard Time at the conclusion of the round. PT providers' reports are scheduled 10 days after the round. If you participate in a PT Express, the report will come out 3 days after submission from the participant. There are 2 types of delivery options, directly through NSI or through a distributor. Distributors can order materials at any time, providing with a payment or instruction from the participant. Also, once available, distributors can order PT Express for the participants. Currently now, we are working through a 5-stage roadmap. And in FY 21-22, we are focusing on pharmaceutical chemistry and materials and microbiological chemistry and materials for both pharmaceutical and non-pharmaceutical testing. Our roadmap also will include in the future biological, clinical, and other areas. Customer service. USP and both NSI provides customer service and email information. And we also provide a 1-800 number. There are different types of questions that may come to different organizations, and we will provide you an example here. In the customer service process, if there is a question related to general chapters or monographs, USP can provide details for that. If it comes or is related to USP methodology or training, USP can help you with that. And for purchasing of service or customized PTs, USP can also support that. NSI supports the navigation of the portal, certain types of data interpretation, participant instruction and information, order and shipping, and payments, and issues related to PT Express and quality control activities. There are some frequently asked questions that are asked by both participants, regulators, and distributors. When are the items available? Items can be ordered within the U.S. customer base within 72 hours of the study. Materials can be shipped within that time also. There's enough material in our studies to perform the analysis twice. The forms of payment for NSI studies and USP studies can be by credit card, bank wire, or PO. Payments must be made in USD. Does this program meet the requirements for both FDA, ISO, and WHO PQ? The program provides objective evidence to be verified by your accreditation bodies as participants in a third-party program. Results can be used for performance for onboarding training, PT, ISO accreditation, WHO PQ, corrective and risk actions, and continuous improvements within and without your laboratory. If you are using a distributor, order your material based on your country's custom clearance processes. The distributors can order material at any time. A distributor must ensure that the study and material arrives at least 5 days prior to the report. If material is lost within your country, it is the distributor's responsibility. Another question that comes from distributors and participants: can orders be returned? The answer is no. Orders that have not been delivered to the participant by the distributor cannot be returned to the manufacturer or USP, as it is impacted by the window of time. Now, are we open for questions?
Thank you, Dr. Charles. All right. Thank you, Dr. Charles, for the very informative sharing. I will take the presenter, right? I'll take some time to give our audience today the updates from USP. And in the meantime, please do keep the questions coming in. I can see a few questions from the Q&A chat box already. We will address them after the updates. All right, let me share my screen. Okay, good. Right, so my session will be update from USP as usual. I'll be giving you an update on the new release, namely the reference standard as well as the analytical material that we have released in October and November month, and then also some compendia updates. Particularly this October, there were quite a few Compendia Updates. So let's go. Here's the table of the new release that we have put it out on the USP store in the month of October and November. So you'll see that there are two tables. The first table on top is the USP reference standard. So in the month of November, we released two new reference standards in the API as well as the impurity category. And then you see the second table on the bottom is the pharmaceutical and analytical material. So quick recap, pharmaceutical and analytical material, PAI, is a new solution launched by USP middle of this year. So it is an analytical material, not a USP reference standard. It is our new suite of PAI that can be used together with our portfolio of official USP reference standards to provide you a comprehensive solution for your research and analytical needs for impurity control. All right, I put in the USP store link at the bottom. So if you would like to understand more, take a look at the USP certificate of the new release. You can feel free to click on the link and then search for the item using the catalog number. Next will be the Usp Compendia Updates. As mentioned, this month, October, there were a few new revision bulletins. I think there were about 3 slides worth, but I'm going to summarize it for you. So on the first slide, these are the new revision bulletins for all these drugs product, I believe, acetaminophen, and I shall not pronounce all of them. So all of them, they have some revision that was posted on 29 October, official on the 1st of November. So in summary, all of them, the test for the organic impurity and the corresponding revision to the USP reference standard session in the monograph has been postponed based on the comment received on the Pharmacopoeia forum. Okay, so I put quick links for you to look at the individual notice. So if you are doing any related work to all these drug products, all right, feel free to click onto the notice and take a look at what has been proposed to be revised. Next, they are the diphenhydramine hydrochloride capsule as well as the same chemical with the different kind of forms, drug in different kind of forms. So there is an update for them as well, posted on the 29th October and official on the 1st of November. So in summary, what was the notice? The notice is that the test for the organic impurity and the corresponding revision to the USP reference standard session in the monograph has been postponed based on the comment received in the Pharmacopoeia forum. Okay? And the last slide will be individual monograph that has some new revision. All right, for the first one, probenone, there was an added dissolution test 24 to accommodate FDA-approved drug product with different dissolution conditions. And then for the second, dibyl products sodium delayed capsule, right? The proposed of this revision is to add dissolution test 6 to accommodate FDA-approved drug product. And then for glycine, it is related to non-botanical dietary supplements. The limit for monochloroacetic acid and specific impurity in the test for related compound has been postponed based on comment received. All right, and then we move on to rotigotine, right? So the notice is that organic impurity and the USP reference standard session of the monographs has been postponed as well. And then lastly, for the quinidine glyconate, right, the purpose of the revision is to add dissolution test 6 to accommodate FDA-approved drug products with different dissolution condition and or tolerance than the existing dissolution test. Okay, so this is the brief summary of the new revision bulletin of the respective monographs. So I put in the link, quick link on the monograph as well as the notice for you to look through if these are monographs that you are using in your laboratory at a regular basis. Do take a look at the notices and make sure that your laboratory is up to date with the latest update from our USP monographs. Okay, and then if you have any questions, I hope these faces are familiar to you. We have seen them for at least 16 times already. So please do not reach out to your APAC representative, your strategic customer development manager, Rocky Chu. He is taking care of our users in Oceania region, Australia, New Zealand, and then myself, I'm taking care of our activities in Southeast Asia. And of course, we have Shin Kim taking care of our North Asia customers in South Korea as well as in Japan. So you have the email address, mobile number. Feel free to reach out to us if you have any questions regarding the USP standard release as well as the Compendia updates. Okay. Now, next part of the update will be the upcoming activities in APAC. There will be some activities coming up in the rest of November. So this week, there will be a USP Quantitative NMR Symposium that will be happening tomorrow, as well as on Thursday, 8:30 a.m. to 12 p.m. Singapore time. This symposium is brought to you by USP China, and then it will introduce the current trend of QNMR around the world and the review of the latest development of adopting QNMR as an analytical technique implemented in industrial, contract laboratory, as well as the regulatory settings. So if you need the registration link, please feel free to reach out to your APAC representative. This event is free for all, and then feel free to register your interest by today because the event is happening tomorrow. And then early December, USP will be hosting an event in collaboration with the APAC COE, and then the title of the event is Securing Medical Products Quality Through the Supply Chain. If you need the registration link, please feel free to reach out to your SCD manager as well. And then, of course, in December, do join us for the last edition of the Quality Hour for the year. So very likely we'll be discussing about the topic of dietary supplements and herbal medicine. Okay, the registration link will go live soon in a few weeks, and then I'll be sure to disseminate the registration link whenever it is live. Now, so we'll move on to the question and answer, right? There are a couple of questions that were submitted during registration. We will address them first. So first question, Dr. Charles, are you able to give an overview of how proficiency testing is done in a microbiological lab?
Yes, can you hear me?
Mm-hmm. Yes.
So similar to solid dose chemistry, microbiological proficiency test is based on a qualitative results. So you may have a positive or negative or a suite of samples that you will have to identify either the organism or you have to identify a negative or positive results or absence of the organism. There's also tests related to sterility that we have, endotoxin, bioburden. There's multiple tests where it is expressed that it's either going to be a negative or positive results. Those results are entered as zeros for negative or 1 for positive during a proficiency test. If the participant is successful, they will identify all of the organisms successfully or the absence or presence of the organisms successfully for microlab proficiency testing.
Okay, I hope that answers the question. We'll move on to the second one. Actually, we have a similar question in the live group chat, live question and answer as well. So how to improve the members who has a Z-score that is not ideal, which is more than 3?
So for a Z-score that is, for those of you who are not aware, a Z-score of 3 means that that participant did either not pass, was not successful, or did not have an acceptable results. Now, it is not a matter of improving, but first you have to go through an investigation to determine why that individual was not successful in the PT. It may be lack of experience with the method. It could be equipment or a variety of other areas through an investigation. It may have to also be retraining for that particular method in the lab, and therefore the laboratory will have to review their training policies, processes, and procedures to help that participant or that member improve their performance prior to initiating a new PT or study.
Okay. Next question, how often do we have to participate in proficiency tests?
So that's a two-part question. If you are ISO accredited or WHO pre-qualified, you have a scope and a time to perform all of the tests within your scope. And the accreditation body will tell you that you cannot get reaccredited unless you perform these specific tests within that specific time. That's for accredited and qualified labs. Okay? The second question is, if you're not accredited or qualified, it's up to the lab to determine how often to participate in the proficiency program. If you have a training program, the majority of labs have multiple individuals perform a specific set of schemes on an annual or biannual basis. So for example, if you have 20 analysts in your laboratory, all of your analysts should be proficient in the methods in the laboratory, but only a select few analysts will perform a proficiency test annually, and some labs rotate through staff to perform proficiency.
Okay, next question. Can we apply bracketing in executing proficiency testing? To which scope or extent do we need to comply for PT? Will doing a risk-based analysis be able to justify the reduced in testing frequency, I think?
Okay, can we apply bracketing to execute proficiency tests? Are you talking about bracketing of the same sample or bracketing of multiple people doing the same sample? I need clarification on that question. So whoever submitted that question, can you please type in the chat area so we can clarify that? To which scope or extent do we need to comply for PT? Every laboratory determines their own scope based on their accreditation body and based on their internal processes, procedures, techniques, and methods. So if you want to determine which scope and what extent to comply, you need to follow up with your PT provider, and your PT provider will tell you what scope you meet the requirements for. Will doing a risk-based analysis justify reducing the test? Risk-based analysis is a means to not only reduce the testing, but a means of performance. But it's not justified to use risk-based analysis to reduce the number of tests, especially when it's related to PT.
Okay, thank you, Dr. Charles. Yes, for the participant that sent in this question, please do help to clarify on the bracketing question that you have sent in, and then so that Donald can help to, address your specific questions. And now we'll move on to the live Q&A. So a few questions. First one, hi, I would like to check for the PT service, will USP be providing the product and the laboratory will be performing their study? Is that correct?
That is correct. USP will provide the material and the instructions. It'll be shipped to your lab, a lab of interest, and the laboratory has to perform the study and submit the results.
Okay. Next, when is the best time to conduct proficiency testing for newly hired laboratory analysts?
So we get that question all the time. So each laboratory has their own policy, processes, and procedure, and each laboratory should have a training program. When you onboard new analysts, it's most likely a three-step process. They will watch you, you will do it together with your analysts, and then they do it by themselves. Upon completion of doing by themselves, a supervisor or manager must authorize or approve that analyst, deeming them qualified to do that specific test. After they've been deemed qualified or authorized, then the analyst should be able to perform a PT because the laboratory has already verified that the analyst can do the test prior to performing a PT.
Okay, thank you. The next question is also regarding less than ideal Z-score. Right. So in the circumstances, if the participant did not pass the PT test or has a low Z-score, what is the best next plan of action? If retest is to be conducted, what and how should it be conducted?
So the next plan of action should be, of course, go through your policies, do an investigation, do a corrective or preventative action, and maybe risk mitigation. Figure out why that individual or participant did not perform well in the test. They may want to repeat another test in that same discipline prior to taking a new PT. Now, if retesting is conducted, how should that be conducted? Most of the time, the same individual using the same equipment and the same method will run the test. Now, at times, we have found that that same individual may fail the test twice. Sometimes the test is not, or individual cannot perform a test. At that point, another investigation needs to be done, and the laboratory can select a new candidate or individual within the lab to perform that test. But you must resolve why the original participant may have failed it the first or second time.
Okay, I hope that answers your question. Next question. How is the assigned value and standard deviation for PT established?
So we use a criteria, and we have both Z-score and consensus value, or assigned value and consensus value. The assigned value is based on in-house test that USP does. We determine the results, and we have a known value or results. Then we submit that material to the participants. The participant develops their own results, and we measure how close or how accurate and precise their results are to USP results to develop a Z-score. The second question to that is, there's also times when we have a consensus value score, which means although USP has done the test, for example, if we do a microbiological test that is quantitative, we allow all the participants to develop their results, submit back their results, and we develop a Z-score based on their results divided by the standard deviation to get a value and establish a value based on the consensus value from all participants. You must have a minimum of 3 or more participants to do a proficiency test. Usually we have 21 to 33 participants involved in proficiency tests at a particular time.
Okay, thank you, Dr. Charles. I think there is a very similar question that you might have already addressed, and then if you have additional input, please feel free. So this question is, in the computing of the Z-score, standard deviation of results amongst participants are taken into consideration. Do you have a minimum number of participants required for each PT scheme?
We require, according to the standard, 3, but our regular requirement is between 21 and 33. Because To receive 95% confidence, which is K equals 2, which is a mathematical calculation, you must have at least 21 participants in a regular PT scheme, but the minimum is 3.
Okay, thank you. That's great. Next question. This is a little bit more, not so much on the technical aspects, but the future plans for USPPT program. So, next, USP has planned to launch PT tests for nitrosamine impurity in pharmaceutical product by using GCMS?
Yeah, so we are planning to do that because that is one of our big topics here at USP. Mm-hmm. And we will be developing that in probably round 2 or 3, which is in the next 2 or 3 months. We have to go through our own internal testing first. We have to receive feedback from our participants. So if you or the participants here want that test to be moved up in the cycle, you'll have to email our representative in Asia or contact USP directly. But there is a plan to evaluate that material via GC as a PT study.
Sounds good. I'm sure to this participant that submit this question, we will be reaching out to you shortly, maybe to understand a little bit more on if there's a specific nitrosamine impurity that you will be targeting in your laboratory methods development. Okay. So next question: do we need proficiency tests on all tests? I mean, does it need to be done on simple test method as well?
If your laboratory is accredited, or if your laboratory wants to confirm the competency and technical capability of the staff, for example, pH as a simple test, Carl Fisher, loss on drying, it would be suggested that the staff be determined to be competent by a third party. Some labs do inter-laboratory testing, which means they make a sample inside their laboratory, and then the laboratory staff will perform the test. But if you're attempting accreditation or pre-qualification, or a regulator is visiting your facility, they may ask you to do a test that's controlled and developed by a third party. So we offer both simple as well as complex tests.
Okay, so we will take one last question. So, Dr. Charles, what is the ideal timeline for PT program of one method?
Timeline as in development or timeline as a completion?
We will wait for the participant to elaborate the question.
Thank you.
All right, in the meantime, okay, so the question is the timeline for completion of one method.
Okay, so when a participant orders a method, they must complete the test within the round cycle. Our rounds are between 30 and 45 days. So if you order the material, analyze the material, and provide the results back, it has to be within that window of 30 to 45 days. If you go beyond that window, your results will not be deemed valid. So we give you almost a month and a half to complete the test if you order it pre-order, or if you order it PT Express, or if you order it within a routine round. Also, let me clarify, if you order directly from the United States, it takes about 5 to 7 days or more of shipping because there are supply chain issues globally. So you will want to take that into account when ordering a study or scheme and making sure that it reaches your lab within the required time that you need it to reach your lab.
All right. Thank you, Dr. Charles, for taking all the questions today, almost all the questions, but I want to assure the participant that submitted the question on PAI, I will be reaching out to you offline after this session to address your question. And then to the participant that would like the presentation slides, yes, we will be making the presentation slides as well as the recording available 1 to 2 weeks after this session, right? So please be patient, and then we will reach out to you with the presentation slides as well as the recording of today's session. And lastly, friends from the Philippines, yes, I will be sharing with you offline the authorized distributor for the USP Proficiency Test for the Philippines. Okay? All right. So with that, I will conclude today's session. So thank you again, Dr. Charles, for joining us in your evening. Thank you for spending your evening with us tonight. And I really appreciate that. Today's session is really interactive, many questions. Do not hesitate to, send in your questions if you have any after this webinar. We will be happy to reach out to you and then address to you any queries that you have. So I hope you find today's session useful, and then please be assured that we are here to help and answer any questions that you might have. Please do keep in touch, and we hope to see you again in our last webinar of the year next month in December. And after this, you will be receiving an email to give us some feedback on how you think about today's webinar. I hope you will take some time to fill up the survey. And then because your input is very important for us to further improve our content of Quality Hour. So with that, I thank you for your time and your participation. I wish you a pleasant day ahead, and please, everyone, keep safe and take care. I'll see you next month. Thank you. Thank you, Donald.
No problem.
All good now. I will wrap up today's session and then send you the statistics for today as well as the questions that were submitted.
Okay, and then I will send you the fly stack, yes. And I will also I need did they want that nitrosamine by GCMSD or just by GC?
GCMS.
GCMS, okay.
Yep.
Good. We were talking about that because we were also talking about alcohol too.